Hi to all. Today I'm going to take you through a fascinating exploration of genetics, specifically focusing on alleles, inheritance patterns, genetic crosses, and epistasis. This poster breaks down key topics in genetics that help explain how traits are expressed. We begin by understanding the basics. Genes are sections of DNA that code for specific traits, and alleles are different versions of a gene. For example, one allele might code for a dominant trait, while another codes for a recessive one. The combination of alleles from both parents determines an organism's phenotype, observable traits, and genotype, the genetic makeup. Let's talk about inheritance using two important types of genetic crosses. Monohybrid crosses involve a single trait. Here, the dominant and recessive alleles interact, and we use Punnett squares to predict possible outcomes. Dihybrid crosses extend this to two traits. We can see how alleles of different genes assort independently during gamete formation, often demonstrated using a 4 to 4 Punnett square. Notice the 3-to-1 ratio in monohybrid crosses and the 9 to 3 to 3 to 1 ratio in dihybrid crosses in F2 generations. These ratios help predict how traits might appear in future generations. Moving on, let's dive into sex-linked inheritance. Traits like colorblindness or hemophilia are carried on the X chromosome. Since males only have one X chromosome, they are more likely to express recessive traits, as they lack a second X to counter the effect. We can visualize this using Punnett squares for X and Y chromosomes, showing how these traits are passed on and why they affect males and females differently. Epistasis is a more complex form of inheritance, where one gene can mask or modify the expression of another. An example would be how coat color in animals depends on the interaction of multiple genes. The 12 to 3 to 1 or 9 to 4 to 3 ratios reflect how gene interactions can alter expected phenotypic ratios. In simple terms, one gene's function can interfere with or suppress the expression of another, highlighting the complexity of genetic inheritance beyond Mendel's original principles. Now, next, we have the chi-square test, a statistical tool used in genetics to compare observed data with expected outcomes. For example, if you conducted an experiment with peas, you could calculate whether your observed ratio of dominant to recessive traits significantly differs from the expected Mendelian ratio. In summary, this poster takes us on a journey from basic Mendelian genetics to more complex inheritance mechanisms like epistasis and sex-linked traits. Understanding these concepts helped us appreciate how diverse and intricate the biological world is. Thank you for your attention. I'd be happy to answer any questions or clarify any points further.